Okay, let's take a look at the chats. Okay, so we have Marius is interested in food microbiology and infectious diseases. All right, so we have two participants who are interested in infectious diseases and who are from microbiology background. That's interesting. Dohan is from Indonesia and has a background in agrotechnology and has a bachelor's research was using amplicon 16S gene sequencing of soil samples. Wow, that's pretty interesting. And that's how you got interested in metagenomics. Let us go to the next person. Prince has studied genetics and biotechnology. And for bachelor's, genetics for my master's. And now I'm doing my PhD, and my interest is in genomics. I'm joining from Benin City, Nigeria. Thank you so much, Prince, for introducing yourself. What about the rest of you? I can see that some of you are still a little bit shy to introduce yourself in the chat box, but it would be great if you could introduce yourself. You can feel free to let us know about your educational background, your research interests, just to get to know the kind of participants with whom we'll be interacting for this session. All right, Muhammad is also is having a microbiology background and is specializing in virology. Okay, great. So we have a lot of microbiology students with interest in infectious diseases. Are you guys joining the session for the very first time, or have you been enrolled in any of the programs before? Is this your first time joining? Do let us know in the chat that as well. And I believe we have just a few more minutes left. All right, Irenaeus Niami, I hope I'm pronouncing that correct as well, is an undergraduate biomedical science student at the University of Cape Coast, Ghana, and is interested in the career of neurogenomics. That's pretty interesting, neurogenomics. I've heard that term before, but not widely heard of it, but that's pretty interesting. Could you please elaborate how you got interested in that as well? Was it as a part of your degree? Did you come across research articles? And for those who are new to the term, feel free to briefly explain that as well. I'm sure a lot of the participants might be interested to know. We also have Dr. Rhoda, who's a microbiologist and whose research interest is in antimicrobial resistance and biocontrol of infectious agent, and is interested in primer design, phylogenetic analysis, and understanding of next generation sequencing. Thank you so much, Dr. Rhoda, for joining today's session, and we hope that you'll find today's session valuable for your research work as well. All right, so Nelson from University of Ghana has a background in infectious diseases and biochemistry. And Nelson is interested in human genetics and is currently working on sequencing human exome to identify SNPs and other polymorphs. That's pretty interesting to know as well, Nelson. And what about the rest of you? I'm looking forward to your updates as well. For those of you who are joined with us for the very first time, we are just catching up with the participants, knowing about what is their educational background, what are their research interests, pretty much that. And feel free to let us know in the chat as well for those of you who have just joined. And we'll be starting with this session in just a couple of minutes. We are waiting for a few more participants to join in case they are running a little bit late. All right, so without further delay, let's start with today's session. And for those of you who have just joined us, again, just a note, feel free to introduce yourself in the chat box. A couple of the participants have already introduced themselves. And along the way of the session, if you have any queries, please feel free to put them in the chat as well. And while we are presenting, we'll take a look at it and try to address all of the queries that you have. All right, so hello everyone, a very warm greeting. Welcome to OmicsLogic. And today we have the open interactive session for the various fall semester programs that we have on genomics and metagenomics. And during this session, we will introduce you to the OmicsLogic Learn portal, guide you on how to navigate it, walk you through our meticulously designed training curriculums for each program, the various training levels, as well as the associated resources. So before we begin with today's session, for those of you who are joining with us for the very first time, let me also take a brief moment to introduce you to the team behind the program. We are a US-based bioinformatics company who is working with multiple academic and commercial collaborators to develop easy-to-use analytical tools, and our mission is to make bioinformatics more accessible. And so far, the training program have been completed by over 24,000 participants from 187 countries in over 300 workshops. And our training coordinators are regularly updating and customizing the curriculum to suit the needs of students and researchers around the world. Now, in order to create a clear path of skill development, we employ the Bloom's taxonomy levels of learning. It begins with students recalling and memorizing facts, progresses to understanding the concepts and applying their knowledge, then moves on to analyzing data and evaluating methods. Ultimately, students reach the highest level of learning where they can design and create their own original work. And now this is achieved through a combination of online courses, training modules, data analysis tools, curated data sets, and interactive mentor sessions to provide theoretical and practical learning in bioinformatics. Now, to further enrich the training curriculum, we also provide real case studies that demonstrate the practical application of bioinformatic methods and omics data in fields such as medicine, biotechnology, agriculture, and even astrobiology. And by using publicly available data sets, these projects allow students to explore and comprehend various research questions and analysis. And the OmicsLogic Learn portal gives students options for analyzing a given data set, which is code independent path or code independent path. Now, if you are someone who is interested in opting the non-code pathway, they will be given directions to access the tBioinfo platform for bioinformatic processing and analysis of data. So this platform includes example pipelines as well as data management and analysis cloud infrastructure to run various bioinformatic pipelines. And different stages of analysis are performed in different sections on this multi-omics platform. We'll be covering about the Bioinfo platform in a short while. And now here are some of the student case examples that I want to share with you briefly. So for the very one example that I want to take is our student, Rithvi Vacha. She was one of our students who worked on assessing the underlying reason for gray matter lesions in multiple sclerotic patients. And she went on to publish her project in Immunogenetics, which is an open access journal that focuses on the genetic research areas of autoimmune disorders such as multiple sclerosis, rheumatoid arthritis, and so on. And further, you also have the opportunity to participate in post-presentation competitions and win exciting cash prizes. And twice a year, we conduct the Omics Research Symposium, which is an international research event that brings together experts from all around the world. And the student projects are not just limited to university students. For instance, Andre Muadano, he was a high school student when he joined one of our training programs, and he presented his neuroscience project at the Regeneron International Science and Engineering Fair, for which he won the Potemkin Prize. Now, this program can also be integrated as a part of the existing university curriculum. For instance, our Python course was part of the Systems Medicine graduate program at Georgetown University. And these projects can also be showcased for various other purposes. So here are some of the instances of how our former students have showcased their projects. So for example, Dilara Taikin, who got accepted to Erasmus IMBRC program in marine biology. Then we had Dhruv Mehra, who is a precision medicine graduate student at Manchester, and Anusha TA, who got accepted to the PhD program in Metagenomics at University of Sweden. Now, faculties and researchers have also collaborated through our programs to publish their research work. And one of our collaboration has been with a team of researchers at Noguchi Memorial Institute for Medical Research at the University of Ghana to carry out computational-based study to understand variants associated with sulfadoxin resistance in Plasmodium falciparum. Now, let's say you are someone with a passion for science and business. It is also possible to pursue an entrepreneurship and business innovation track like a team of industrial researchers from a biotechnology company in Nigeria did. So they worked on developing a microbiome test kit to improve the fertility of women in Nigeria and improve their chances of carrying their babies to term. Now, these entrepreneurship tracks are not just pursued by researchers, but by students as well. For instance, a group of undergraduate and postgraduate students worked on developing an early detection kit for Alzheimer's disease. So now you can take a look at all of these student projects and researchers project at our OmicsLogic Learn portal. So to view these, let us head to the OmicsLogic Learn portal. And for those of you who are joining with us for the very first time, let me share the link in the chat box so that you can create an account. Those participants who are interested to take a look at what are the various courses, what are the various training programs we have, and what are the various student projects that have been completed so far, you can take a look at all of these collections on our website. So let me just give you a brief demo on how you can sign up. So for those of you who are starting for the very first time, when you head to the link, you will see a create an account option like so. Click on that, and there it will ask you to enter your name, email ID, password, and click on next, and it will ask you to enter a few more details. Once that is done, you will be able to log into your account. Or if you already have an existing social media account such as Google or Facebook or Apple account or GitHub or Twitter, you can also log in just by clicking on that. Now, if you are someone who already has an account on the portal, then all you can do is enter your existing email ID and password and directly sign in. So I'll just pause for a couple of seconds. For those participants who are creating an account and want to log in, they can do that. And along the way, if you're facing any technical issues while signing up, please feel free to put that in the chat as well. And our technical team will be able to look into that. So I'll just pause for a couple of seconds and monitor for your response. For those of you who already have an account on the portal, please put it done in the chat box so that I get an idea. And for those of you who are creating an account and once done, put a yes in the chat box so that I'll know that as well. All right, so I can see that a lot of the participants have created an account. Perfect. Arina says done. Jyotsna says done. Maria says done. Landry says done. All right, Mary, who just joined, we are just signing up on the OmicsLogic Learn portal where I can take the participants and showcase the various courses and training programs that we have. Let me quickly share the link in the chat box so that you can start with it. Right, so without further delay, I'm just going to sign in with my existing account. All right, so once you sign in, the very first interface that you're seeing is the courses page, where you will be able to see a list of collection of courses that we have on the portal. So if you scroll down below, you will see a search bar that says looking for a specific course. And here, if you enter a key area of research interest, or let's say a specific field that you want to specialize in, then you can enter the keyword and it will show you the various courses and example projects that makes use of it. So just as an example, I'm just going to enter the keyword genomics, which is associated with today's session. So as you can see, it shows up the relevant course, which is on genomics, as well as a wide variety of example projects ranging from infectious diseases to oncology and so on. So you can explore this at your own pace and take a look at the various courses and example projects that we have on the portal. And now, once you sign in, you also have to make sure that your profile is updated. So how do you do that? You can head over to the blue bar on the top that says welcome back with your email ID. Click on that and that will directly take you to your account settings. And here, if you click on the profile button, You will be able to edit all the account details that you want to get updated on your profile, which includes your profile image. Make sure that your name is in proper spelling and proper capitalization. And you could also link your social media handles as well. As you can see in my case, I've linked my LinkedIn account and my org ID. And add a brief bio about yourself as well so that we know who you are, what is your background, what are your research interests. In my case, I've mentioned my educational background and research. So once that is done, click on update and that will be getting updated. So that was all about that I just wanted to discuss with you. So now to get started with the fall semester programs session, I'll pass on the stage to my colleague, Sonali Kari. Over to you.
Thank you so much, Srigori. Can you confirm if you can see my screen and if I'm audible?
You're audible and we can see you.
Perfect. A very warm good morning or good evening in whichever part of the world you are. I'm Sonalika Ray and I'm the Bioinformatics Project Manager at Pine Biotech. Today, I'll talk about the various fall programs that we are offering. I'll talk about each one of the programs very briefly, what it includes and how the program would be conducted. So let's go by each one of these programs one by one. So for the fall semester, we have Introduction to Bioinformatics, Genomic and Metagenomic Data Analysis, and also Biomedical Data Analysis using Python. So the goal of the OmicsLogic programs is to provide training in bioinformatics, which will enable independent research guided by mentors and peer example, and also develop a growing community with shared passion for data-driven research and appreciation of citizen science, enabling students, clinicians, and faculty of all the backgrounds to develop novel and independent research using latest technology in life and data sciences. The training program would offer personalized mentor guidance, expert videos, live support, hands-on tools, and certification as well on successful completion of the program. Now, let's take you through the very first program, that is Introduction to Bioinformatics. So for those who come from a non-bioinformatics field, let us quickly understand what is bioinformatics and how do we really use it. So bioinformatics is the science of both storing lots of complex biological data and of also analyzing it to find new insights, which we have to use in many different ways for analysis. And today's biomedical research isn't simply about publishing one set of results and hoping other researchers read it. It's about finding patterns in all forms of data, linking everything that is out there to provide new insights that we can only spot if we can see the big picture. Bioinformatics lets us bring together the data from lots of experience in one place so we can ask those big questions and find the answers. And such data from different sources is available rather than availability of such rich sources of data changes the nature of the biological research itself, as it is rapidly acquiring the character of a data science. Now, who uses bioinformatics and who is it for? The answer might have been statisticians, computer scientists, or engineers. But the truth is that more and more biologists, as well as clinicians, are now expected to understand and handle their own data to publish and succeed in basic and translational research. In fact, biology itself is rapidly acquiring the character of a data science. So as biology is rapidly acquiring the character of data science that I just mentioned, billions of data points on genes, proteins, and other molecules are compiled in large files and systematically studied. And analyzing this data leads to more knowledge and understanding about living organisms, which includes human health, biotechnology, crops, and livestock. So this training program will give an overview of bioinformatics for understanding multi-omics data analysis for students, biologists, and clinicians, and the influx of data that is associated with these methodologies and data-driven research makes bioinformatics for life sciences as prominent as the emergence of computers is for the tech industry. Now, the field is based on high-throughput technologies that generates billions of data points on genes, proteins, and other molecules which characterize biological systems and phenomena. And this data that is compiled into the repositories and can be systematically studied to understand, interrogate, and establish trends for many applications, which includes phenomics, genomics, epigenetics, and also transcriptomics, and what is missing here is single-cell transcriptomics. Now the question is such data available? Yes, and one such repository is maintained by National Center for Biotechnology Information, which has a collection of peer-reviewed articles, biological datasets, and projects on various topics. And as you can see, in the past several decades, this resource has been growing exponentially. There are over 80,000 publicly available datasets in just one part of NCBI that is called GEO, which stands for Gene Expression Omnibus. And this is just one simple example of a data repository which contains datasets on thousands of genes in each sample. But in the program, we'll explore more and more of such repositories and understand what type of data is stored in these repositories, how we can extract it and make the most out of it. Now, the program is designed to highlight the role of big omics data that is shaping modern biomedical innovation, where molecular data is used for precision diagnostics, big data integration, and machine learning that is transforming personalization of treatment selection and innovative algorithms that allows for unprecedented capabilities with drug discovery, design of large molecules, and even drug repurposing. The program will cover some of the important technologies and its applications in today's world. The participants will be able to access the bioinformatics resources along with expert and mentor support to get help with the learning process that will help you learn the transformation of biology to data-driven biology. And this was about the first program, Introduction to Big Data Bioinformatics, where we'll cover these aspects of different omics analysis. We'll get you introduced to these different omics analysis, which includes a genomic data analysis, transcriptomic data analysis, metagenomics, single cell. Now let's discuss about the other program, which is genomic data analysis. So whether you are just starting your journey or looking to expand your expertise, this program is designed to provide you with the skills and the knowledge that is needed to excel in this rapidly evolving field of genomics. So there are 3 different levels of the program that I'll take you through when I go through the program pages of these programs. But to talk about the topics, I'll divide them up into beginner, intermediate, and advanced level. So at the beginner level, you will develop a strong foundation exploring bioinformatics and also understanding next generation sequencing, data exploration, and also you will learn about sequence alignment and more. So when we talk about sequence alignment, from there, I get this picture of the workflow that is very important for us to understand. So we have the sequencing reads. We need to understand how to take it up for quality control, and then we align them up. And after aligning them on the reference genome, we want to understand how to look for these specific variants, and then finally understand what these variants actually are, or what type of variants these actually are. So we need to annotate them. What are the different types of files that are included at all of these different steps, and what all of these different steps includes, and how do you get to know about a specific type of variant and annotate it effectively is what the program is all about. So at the intermediate level, we will take you deep into the intriguing topics such as variant calling. We'll process the variant calling pipeline. We'll look at the results. We'll try to interpret these biological insights out of the results that we get. And we'll also discuss about, Shrigori, can you take care of this? Someone is requesting to record the meeting locally. We can share the recording of the session with you Bistan. Sorry if I am pronouncing your name not correctly. We are also live on YouTube, so you have the link from there. I'll deny your request at the moment. Thank you. All right. So from the variant calling, we'll also learn about copy number variation analysis, machine learning, genome-wide association studies, and also briefly touch upon the ethical considerations. And for those seeking advanced knowledge, we'll delve into population genetics, DNA sequence analysis, VCF file analysis, functional annotation, and also we'll discuss about these real-life case studies on SARS-CoV-2, breast cancer oncogene variant calling, and also Ebola. And by joining this program, you will gain practical hands-on experience, learn cutting-edge techniques, and also unravel the mysteries that are hidden within the genomic data. So be prepared to unlock a world of exciting possibilities and make significant contribution in the field of genomic data analysis. Next up is metagenomic data analysis. To unravel the microbial community, to explore the limitless potential of metagenomic data analysis through our comprehensive training program that is designed for beginners, intermediates, and also advanced level practitioners. So whether you are starting your journey or seeking to expand your expertise, this program equips you with the skills and the knowledge to excel in this dynamic field. Now let's get back to understand where did the metagenomics field come up from. So in the 1800s, the link between disease and the microorganisms was established, shedding light on isolated bacteria functions. And today we understand that the human microbiome consists of structured communities forming biofilms with complex interactions aiding ecological stability. And advances in metagenomics enables the study of the vast majority of uncultivable microorganisms within their natural environments. So the course covers bioinformatics analysis of metagenomic sequencing data, which includes genomic sequencing technologies, data utilization, analysis tool, as well as real-world applications. So since the 1900s, microbiome studies have made significant progress, and notably through initiatives like the Human Microbiome Project that enables metagenomic sequencing advances, allows categorization of microbiota in various human organs, presenting opportunities as well as challenges in identifying their roles in organ health and disease as well. So in the program, we will learn about these different types of metagenome sequencing techniques through different types of data that is generated. We'll majorly focus on 16S metagenomic sequencing and the data generated from it, such as OTU sequences that is annotated by mapping onto a reference database, such as the Ribosomal Database Project RDP, Green Genes, or the Silva database. And such databases contain taxonomic information for the domains of bacteria, archaea, and eukarya as well, that is based primarily on phylogenies for small subunit rRNAs, which include 16S prokaryotes and 18S for eukarya. Now, based on the mapping of specific regions amplified from the 16S RNA gene in the small subunit of the bacterial ribosome, OTUs can be assigned to belong to a taxonomic class of bacteria, yielding a proportionate view of the community. And each community can then be studied by comparing with others or between similar communities in different conditions or individuals. And such analysis includes an in-depth understanding of data preparation, reference databases, and analytical approaches to answer specific questions based on solid evidence. Importantly, sequencing data that represents only one type of microbial omics, with many more becoming increasingly available. So there are many questions about the role of microbiome members and the communities that are now being answered using next-generation sequencing genomic data, as well as mass spec approaches for proteomics and metabolomics. Indeed, the next phase of microbiome research lies in the ability to integrate multi-omics datasets that combine NGS, mass spec, and structural data with phenotypic information about the overall changes in the community and their environment. And in the program, we will go through these three different pipelines that I'll just briefly touch upon. The first one is the taxonomy classification. Then we have the data2 pipeline that denoises and corrects errors in amplicon sequence data, providing high resolution error-free sequence variants as output. And next, we will also learn about KIME2 pipeline, which analyzes and processes microbial community data, offering diverse outputs like taxonomic assignments, diversity matrix, and visualizations. And behind many of these methods are scripts that are written in popular coding languages such as R and Python. And these are also explained in the tutorials that you will have an access to as a part of the program that I'll just take you through briefly. It includes beginner tutorials on data importing, preparation for analysis, and also standard visualization of the processed data, and more specialized packages with publication-ready outputs that will also be covered in more advanced lessons in the Metagenomics coursework and the case studies that we'll take you through. We will also learn about the basics of functional metagenomics that explores the genetic potential and metabolic activities of microbial communities in a given environment. So what kind of projects can be studied using metagenomics sequencing or metagenomic data? In the program, we'll go through these number of applications that includes skin conditions, food sensitivity of the gut, autoimmune disorders, cancer, antibiotic resistance, and also we'll try to understand the microbiome that is involved in drug resistance and even in ecological studies. These are just few applications that we'll touch upon that I just mentioned, but there are more such case studies that we'll cover in the program. In this program, we'll also learn to find and analyze data from such repositories as the Human Microbiome Project, which includes a data set that is called the American Gut. This citizen science project has a lot to offer for those interested in basic microbial composition of different types of samples from healthy and mildly sick individuals. So that was about metagenomic data analysis. And the next program that is being offered this fall is biomedical data science using Python. So the demand for skilled data analysts and scientists who can derive meaningful insights from biomedical data has never been greater. And that's where our online mentor-guided training program on biomedical data science using Python comes into play. In today's rapidly evolving world of biomedical research, the generation of big data has become the norm. Every day, vast amounts of valuable biomedical data is being produced, holding the potential to revolutionize our understanding of diseases and pave the way for groundbreaking medical discoveries. However, the sheer volume and also the complexity of this data presents a significant challenge, and that's where data analysis comes in. So this comprehensive program is designed to equip you with the knowledge and the skills that you need to excel in the field of biomedical data science, and through a combination of practical assignments, live workshops, and online resources, you will embark on a transformative learning journey that will empower you to unlock the potential of high-throughput biomedical data. So now what the different types of levels will include. The beginner level training program serves as a very solid foundation in Python programming and its applications to biomedical data analysis. We understand that not all of you come from a bioinformatics background. That's why we'll also introduce you to the different types of biomedical data, omics data that is being generated, and what the structure of this data looks like. And then we'll get started with the very basics or the fundamentals of Python in the beginner level. We'll start by understanding the basics such as data variables, control structures, and also we'll discuss about functions and modules. Then we'll dive into data processing and cleaning using powerful libraries like NumPy and Pandas. Armed with these skills, you will move on to the expiratory data analysis leveraging visualization libraries like Matplotlib and Seaborn to uncover patterns and relationships that are hidden within your data. Additionally, you will gain an understanding of dimensionality reduction techniques and machine learning using scikit-learn. And as you progress to the intermediate level, you'll deepen your understanding of clustering and advanced machine learning techniques specifically tailored for biomedical data analysis. You'll explore clustering techniques like k-means, hierarchical clustering, spectral clustering, using Python clustering packages like scikit and scipy. And you'll also learn about model validation techniques and evaluation matrix for supervised machine learning models. Furthermore, you will delve into the advanced machine learning techniques like decision trees, random forests, SVM, LDA, UMAP, and apply them to the real-world biomedical problems. And last but not the least, I want to discuss about the artificial intelligence, where in today's data-driven world, AI that is powered by technologies like ChatGPT that is all around the place has revolutionized the way we approach scientific research and data analysis. And as learners, you have a remarkable opportunity to harness the power of AI to unlock new insights, streamline complex tasks, and also accelerate your learning journey. AI, particularly the code interpreter that I'm very fond of, can provide assistance by generating code for complex analysis and also automate routine tasks. For those who are biologists and are not hardcore programmers, this is a blessing in disguise. Additionally, it can produce insightful visualizations such as heat maps, dendrograms, and also other plots to help you visualize and interpret your data very effectively. We are committed to equipping you with the knowledge and the skills to leverage these AI capabilities as well in your academic and research endeavors, which can fasten up the pace of your research and also definitely have these complex tasks done in a less time. Together, together we'll embark on a transformative learning experience to explore these endless possibilities that AI offers in the realm of biology and beyond. So that was all about these different training programs that I wanted to discuss. And now I'll quickly share my screen and take you through the OmicsLogic Learn portal, the resources that we would be covering or the resources that you would get an access to as a part of this program. So as Sri Gauri already took you through the OmicsLogic Learn portal and helped you understand where your profile is, where the courses are, I'll just take you through the specific resources that you'll have an access to as a part of the program and what all you need to cover. So for those who are interested in Introduction to Big Data Bioinformatics program, they can visit the program page. I'll just show how to visit the program page or how to reach the program page. So on the portal, you have a courses tab and then you have a programs tab. Under the programs tab, you have all the programs that are currently running. So you need to here look for, you can also type bioinformatics. You simply need to click on the specific program page of your interest. And because I already have it open, I won't reopen it again, but I'll just show you how it's done. Once you have the access to a specific program, the program page will also appear on your profile as a button so that it's easy for you to access it. So if this is your profile, on the extreme left, you have the badges here. And if you click on it, you will be redirected to the program page. A lot of program users and students have doubts on how to get the access to the program page. So that was just a brief demo on it. All right, now coming back to the program page. Let's now discuss about the sections of the program page so that it's easy for you to just go through all of these resources. On this program page, you will have all the lectures and the resources put together. So we have 3 levels, as I mentioned: beginner, intermediate, advanced. there are different number of sessions for each level. It depends how much time you have and also what type of expertise you need for under the specific type of program. So for the beginner level, we have 6 sessions. Intermediate, we have another 6 sessions. And advanced, we have 12 sessions. So in the beginner level, we'll introduce you to the very basics of molecular biology and bioinformatics. Again, the idea here is to introduce you to the field bioinformatics for those coming from a non-bioinformatics field. You might be a biochemist or a microbiologist, and we have also noticed interest of medical students in the field of bioinformatics. So this specific session will help you get started. Now, if you look at the resources for a specific session, let's look at the breakdown. You have the session objectives, you have the session outcomes, and then for the specific session, you have some parts under it. There could be 2 parts, 3 or 4 parts under each session. And for each part, you have a session video, that is a session lecture that you need to watch. And you also have the associated lesson that you need to complete. And you also have a keywords help book that you can go through and the additional reading material. Because this is just the very beginning of a session, that's why you don't have the practical hands-on. But if you scroll down and explore other sessions, you can see we have more parts here. And also you will get a link to the demo pipeline and the practical hands-on. For practical hands-on, you have the data. So if in a session, the mentor is taking you through a practical, let's say. So the data set that the mentor used, you will have the link to that data here. And you also have a link to the demo pipeline that I'll just take you through. And you also have to go through the lesson that you have mentioned here and some additional reading material. Now, apart from watching the lecture, you have to complete the associated lesson. Now, where is the lesson actually based? So under the courses tab, you have these various courses and you also have these example projects, or I can call them case studies. After completing a coursework, it's very important for you to understand how a specific research question could be answered with the skill set that you gain through a coursework. So for that, you need to go through these case studies as well. That has been structured well in the program that will help you give a wholesome experience, that is from learning about these specific concepts to also having a practical hands-on experience with these case studies. So as I was talking about introduction to bioinformatics, along with the program, you have to go through the Bioinformatics Coursework too. That is the first course on the portal, that is Introduction to Bioinformatics. Now the courses on the portal are structured or placed in such a manner that under each course, you have several lessons that you need to complete as a part of the training program. And on the training program page, for each of these sessions, you have 1 or 2 of these lessons covered that you need to complete. So you have a structured way in that you have to follow. You watch the lecture, you complete some of these lessons. Just a second. All right, I needed a quick water break. So you can see that you have these lessons under each coursework. And for the score for that specific session, you need to complete a lesson. So that is how these lessons on the program page are given. Now let's look into the lesson. So what is present inside the lesson? Inside the lesson, you will find the concept that is given. You'll also find some lectures or mentor videos, very short mentor videos that you have to go through. And also you will find the assignments and also quizzes that you need to complete in order to complete a specific lesson. So that is how a lesson is structured. In the Introduction to Bioinformatics coursework, as I mentioned that in the training program, our goal is to introduce you to the field bioinformatics and also to talk about different types of omics data, be it genomic data, transcriptomic data, metagenomic, or single cell or epigenomic data, so that you have an understanding of what the field bioinformatics is all about. And if I now proceed ahead with the next program, that is genomic data analysis. Just a second. Yes. All right. So if you proceed ahead with the next program, that is genomic data analysis, you'll have the sessions given in the same order, a beginner, intermediate, and an advanced level. And for the genomics training program, you have to take up the course that is genomics. That is course 3, genomics, right? You need not navigate the course like I did under the courses tab. You have the links of each and every lesson of the course on the program page itself. I'm just showing you the main source of it so that you know where the specific coursework or the lesson is placed. Now, in the genomics course, we are taking you from the very basics of genomics. Then we're discussing about the next generation sequencing techniques, types, what type of genomic data do we get, different types of genomic data, all right, be it targeted data or be it the whole genome or whole exome data. And after discussing about the types of data, we discuss about sequence alignment, phylogenetic analysis, variant calling, mutability analysis, differential mutation analysis. And then in the coursework itself, you can see that some of these lessons have the Learn batch, while some of these lessons have the R-code batch. So also while discussing the concepts and exploring the practical hands-on, we'll also take you through the R code blocks to understand and learn how R can be used as a computational language to work with genomic data and perform different types of analysis. Now, as I mentioned about these different types of analysis that we would carry out for the practical experience, where do we actually do it? So for those who are new and have joined the session for the first time, for them, you have two resources. One is the OmicsLogic Learn portal to learn about these different types of concepts. And then you have the Bioinfo platform that you use for your practical hands-on. So this is the educational reservoir, and then you have here the practical hands-on Bioinfo platform. On the platform, you have different areas of analysis: transcriptomics, genomics, single-cell transcriptomics, metagenomics. Then you have machine learning here as well. Here you have GWAS and phylogenetic analysis too. Now, because I'm discussing about the genomic data analysis program, so I'll just highlight the major analysis that we're going to cover under this program. And for this program, we would be covering variant calling that is given here, phylogenetic analysis, and GWAS as well. Now, if you can see, there is a drop-down button in front of each and every main pipeline. That is actually a demo pipeline or an educational pipeline that is based on a case study on the OmicsLogic Learn portal. So I'll just quickly process this demo pipeline to show you how you can use the demo pipelines for learning about these different tools or algorithms that could be used for analysis. And then we look at the results, the outputs that we get from the pipeline, and we try to understand how do we interpret the biological insights out of the results that we obtain by processing a pipeline. Here the data has already been uploaded at the backend, and I'm just processing the pipeline to show you how easy it is to process a genomic data analysis pipeline for variant calling. And you can also do that, and what results we get and how do we interpret. This is just a demo, and I just wanted to introduce you to these different resources. So I'll not get into much depth of it right now and explain you how to interpret it. We'll take it up in the sessions, obviously. Meanwhile, this loads, let me open the chat box and I can see there are some comments here. Srigori, the comments about certification and recording, please address them. You can share the link with them. And also Srigori will share the recording with all of you on your email once it's processed. So don't worry about it. For all the technical issues, you can reach out to Mr. Ujasvi Datta on dev@pine.bio. If you're trying to sign up and if you face any issues, he would be there to assist you. And okay, we have a question by Ratnadi. Is it possible to analyze the data of dementia patients using data-to pipeline, or can we use dendrograms? So Ratnadeep, it depends. If you have RNA-seq data or genomic data, you can definitely perform hierarchical clustering on it and have dendrograms and try to understand how the dementia patients separate out from the controls, right? But when you're talking about data too, it completely depends if you have the gut microbiome or a specific obviously, if it's dementia patients, it has to be the microbiome from. So there's a gut-brain axis component or concept that you might have heard about. If not, we can discuss about that as well. So if you have the microbiome data for the dementia patients, then it would actually be very interesting to understand the microbial composition or the dysbiosis in dementia patients, because it's well known. And also we have worked on such neurosciences based projects on Alzheimer's or Parkinson's, where we have tried to look at the microbial compositions and try to understand which genes they affect or which pathways they are affecting eventually based on the substrate the communities are producing. So yes, we can process a data-to pipeline as well to understand the data of dementia patients. But it obviously has to be a 16S data. Maybe that would be the best if you have to process a data-to pipeline on our platform. Because on our platform for data-to analysis, you need to have a 16S data. And there are various repositories that you can explore to get the data for dementia patients that we can discuss in the program. How do we get course certification? Mohammed Moez, Srigori will let you know about it. Once the program is completed, you get the certificate. I'll also show you towards the end. What is the best pipeline to analyze mpox data? Landry, again, depends what type of analysis you want to do. We can't pick a pipeline just while thinking what the best pipeline could be to analyze this type of data. To pick a pipeline, it depends what type of research question do you have in your hand. Atifa and okay. Yes, we have the recording. We share the recording with you. And for certificates, Srigori has addressed it. And also, Ratnadeep, if your question was answered or if you have any other question, you can please put it in the chat box, or you can also unmute yourself and ask.
Good evening, ma'am. Actually, my question is, as you mentioned about the data to pipelines, okay, to analyze if we have the specific 16S sequenced microbiome data sets. So my question is basically, as we are focusing on the gut-brain axis, in the gut-brain axis, if at the time of dementia, if it is affecting the thalamus section or the hypothalamus, so is it mandatory that we have to be having that again at the same time to check out how much it is degenerated or how much it is affected? Can we calculate it using these data to pipelines or dendrograms, or can we use heat maps?
We can use all three of them. If we have the access to the 16S microbiome data, we can process a data to pipeline to understand the composition and the affected genes and the pathways as well. And if we have the RNA-seq data, we can definitely proceed ahead with the machine learning approach and try to understand through dendrograms or heat maps which genes are affected, and then try to understand the pathways that are getting affected.
Okay, so to identify that pathways, sorry to interrupt, to identify those pathways which is being affected, do we need like markers like red fluorescence protein, RFP or GFP, so that we can analyze using machine learning or dendrograms? So will that
Yes, we
affect that
definitely need the
RNA sequence
markers.
data? Like, does that biomarker will affect it? Like while we are analyzing using these data to pipelines or heat maps and dendrograms, does that biomarker will has any effect on these data set which you're trying to
Yes,
analyze?
definitely. When you want to do pathway analysis, you have to first of all look at the biomarkers, and from the biomarkers only you get to know about the specific pathways that would be affected. Secondly, you're trying to mix these two concepts. Data 2 is an entirely different type of analysis that could be done on microbial data. The type of data here is different. All right. And if you
Okay.
want to bring in the concept of biomarkers while understanding how the microbial composition affects specific genes or your biomarkers, and then you try to understand the pathways, that is functional metagenomics. And if you want to understand with the RNA-seq data, how or what specific type of genes or biomarkers you say affect a specific pathway, that is with RNA-seq data, right? So
Okay.
these three different types of data analysis is possible, but again, it depends what type of data do you have from your lab, or you want to do a project based on data that is publicly available.
Okay, ma'am. Thank you.
Welcome. And also, if you have a specific research proposal and you want to discuss further how to do it,
Yes.
and what should be the methodology that has to be used, and what type of results you will get, because this is just a very broad discussion. If you want to get more into its depth and try to understand which specific plots you will get and what specific patterns do you want to look for that, we can have a one-on-one call where we can discuss your project objectives and guide you accordingly.
Okay, ma'am. Surely, like if I can get the like email or the contact info and also that we can have one-on-one discussion for it.
For any discussions based on projects, you can reach out to me on sonalika@pine.bio. And also for any questions, you can also reach out to communications@omicslogic.com.
Okay, ma'am. Thank you so much.
Okay, thank you. And this is what the genomic data analysis pipeline result looks like. This is JBrowse 2 version that is linked with the genomic data analysis pipeline. And here you have all of these samples, and you just select the reference sequence and zoom in a little bit. And I can here look for the gene of my interest. This is a breast cancer pipeline where we have these patients who are affected with a specific type of breast cancer, and we have some control patients, normal patients not affected by breast cancer. And I'm interested to look at the variants here. So this is the reference genome. I can just zoom in more and look at the sequence. I have the option here to just look at the nucleotides or amino acids. And if I zoom out a little bit, I can look at the CNV, and the interpretation is quite lengthy, but we will take it up in the sessions. So that was a very brief overview of the variant calling pipeline. And similarly, we have a pipeline on phylogenetic analysis that we'll take it up, where we'll try to understand how we can take these sequences, perform multiple sequence alignment, and then carry out phylogenetic analysis. How do we get these different types of trees? How do we interpret the results from them? Because time is a constraint for this webinar, I'll not go in much depth right now, but we'll take up all of these different points of analysis through the T-Bioinfo platform. And also, we will learn how we could use the open source tools and softwares to carry out all these different types of analysis, and definitely learn how code could be used to take up all of these different types of analysis. So that was about the genomics program. I'll just close these two tabs so that we're not confused. And now let's discuss about the metagenomic data analysis program. So for the metagenomic data analysis program, we again have these 3 different levels: beginner, intermediate, and advanced. And for this, we have to go through the metagenomics training program. One thing that I skipped to mention before when we were discussing about these 2 different programs, if you type a specific keyword in the search bar like genomics or metagenomics or bioinfo and bioinfo and click on project, you'll get the list of all the case studies or the projects for that specific topic. So if it's metagenomics, we have these 4 case studies here that we'll explore and go through. So you will have these 4 case studies here. Okay. Now in the metagenomics coursework, again, we have the concepts covered, and also we have the R code block lessons that we will go through. So as I mentioned, in metagenomics, we have these 3 major pipelines that we will go through. The first one is for taxonomy classification, and then you have the data to pipeline and the kind to pipeline as well, to understand alpha diversity, beta diversity, and other core matrix and functions as well through kind analysis. You all can create an account on the OmicsLogic Learn portal, and after 24 hours of it, you will get the access to the Bioinfo platform. If that does not happen, you should reach out to us on communication@omicslogic.com, and we'll give you your demo credentials if you want to explore and see how these different types of pipelines work. And as a part of the training program, you will get the access to the Bioinfo platform, where you will learn how these pipelines are processed, what tools are used, what parameters to select, and what results you get and how do you interpret them. And before you process a pipeline, it's very important to understand which public repositories have these specific types of data that you can extract, and how do you clean up your files, or how do you process your data to be ready to process these pipelines, and when the data is ready to upload and process these pipelines. So that was about metagenomics data analysis program. And now it comes to biomedical data science using Python. So for this program, we have these 3 levels again: the beginner, intermediate, and the advanced level. And on this specific program page of biomedical data science using Python, you'll also find the link to the Google Colabs. Now we will learn the code of these different types of analysis using Python through 2 different ways. The first one is definitely the coursework. So you have these 2 courses on the portal. One is Python course 1 and another one is Python course 2. So for those who opt for the beginner level or the basic level, can just take up course 1. But for those who opt for an intermediate or an advanced level, they can take up course 2. Now the course 2 will take you from a very beginner level to an advanced level, where you will understand from the very basics, that is how to install Python or how to install different libraries and packages for Python on your system, how to install Anaconda and the required files to process the Jupyter notebooks. And also then you will learn about how to import data, how to import different types of data, how to normalize data, how to perform dimensionality reduction, apply statistical techniques, and also you will learn about different machine learning techniques, which includes unsupervised analysis, supervised analysis, and also you will learn about deep learning and network analysis. Now inside the lesson, I'll just show you one of these lessons. Inside the lesson, you'll learn about the concept of a specific topic. So if it is data normalization or log transformation, why is it important? Why is it required? And then you'll also learn about the code block, and you have the explanation of the code block given as well. The libraries or the packages that are used, what do they do? Why are they used? What type of analysis can you expect from them? What type of results do you get from a specific code block? And for your practice, towards the end of the lesson, you have a practice code step where you have the link of a Google Colab that you need to open and practice the code block that is given in the lesson. So when you practice through Google Colabs, make sure that you copy that collab and then you can practice your code block. Along with that, you also have a session-wise collab that you can practice the codes with. We refer collabs because if you are working with the code block and you get stuck in between, you can reach out to us with the link to your Google Colab and we'll help you rectify the errors that you might get in your code. So that is the reason why we work with Google Colabs. And also you have the links here that you can refer to. So that is how we plan to teach you Python for different analysis at these different steps. Now comes a point where you have to understand how to register for the program. If there are any questions, you can please put in the chat box. Ratnadeep, we can discuss about it. We can discuss about the research objective, and then we can discuss the terms of the platform accessibility for processing the pipelines. All right. Okay. I'll just simply log out first so that I get the interface. All right, so now once you're on the OmicsLogic Learn portal and have signed up and you want to enroll yourself for a specific program, you can simply open up the program page. And if you scroll down, you can see 3 different levels mentioned here: beginner, intermediate, and advanced. And in whichever level you are interested to pick, you can simply click on the Buy Now button. You can reach out to Ms. Sri Gaurian on communication@omicslogic.com. We have these codes that you can use for getting a scholarship to enroll yourself for the program, and Sri Gauri will assist you on getting the access to the code and then proceeding ahead with it. So you can pick the level of your interest, and we have mentioned here what all access you will get as a part of the level. And it depends, first of all, based on the length. So beginner level, you get the access of the program for a month. Intermediate is for 2 months, and advanced level is for 3 months. And also when you complete the advanced level, you get an opportunity to work on a capstone project. And if you have a research question in your head already that you want to work on, like Ratnadeep has a research question that he mentioned. So if you have a research question that you want to work on, we can discuss that. And we can work on a mini project sort of thing that you can use to submit in your college or so. So an advanced level will give you the access to work on a capstone project. And if you want to work on a research project after completing the training program, you also have that option, because once you complete the program, you understand all the concepts that are required to work on a project, but then you need just the directions on how to work on the research project, how to pick up a research question, what data to take, what pipelines to process, what customizations you need, and then finally the results that you get, how do you effectively interpret them and take it up for a publication. So that is also an option. After completing the advanced level training, you can work on a research project that we can discuss. So that was about different levels and how do you register. For those who are in different parts of the world, you can scroll up and here is a button just next to the log out or the login option. And you can just change your currency. For example, I'm in India. So if I change it to INR, I'll be then able to look at the cost of the program in INR. So that would be a help to you. That was all that we wanted to discuss about the program, what access you will get through the program, and also how do you register yourself for the program. If there are any questions, you can put in the chat or you're free to unmute yourself and we can discuss.